Welcome to Creative Diagnostics channel. This video we are going to talk about the GABA release from astrocytes in health and disease. Astrocytes are the main glial cells in the central nervous system. Although they are non-excitable, astrocytes perform a wide range of roles, such as maintaining extracellular ion balance, clearing neurotransmitters, regulating synapse numbers and modulating local blood flow. They are able to detect neuronal activity through various membrane-bound ionotropic and metabotropic receptors, including those for neurotransmitters like glutamate, GABA and ATP, which allows them to dynamically adjust their functions according to physiological needs. Activation of these receptors on astrocytes can trigger transient increases in intracellular Ca2+ and Na+, which often spread through the astrocytic network as waves. Previous immunohistochemical data showed low levels of the GABA-synthesizing enzyme glutamate decarboxylase, GAD, in astrocytes, indicating that the intracellular GABA concentration is too low for nonvesicular GABA release. However, recent studies show that astrocytes in various brain regions can synthesize enough GABA, partly through GAD-independent pathways, and can also release GABA. Additionally, there is evidence that both processes are strongly regulated under pathological conditions. The GABA has been considered to be synthesized mostly by neurons. This conclusion was based on biochemical data, suggesting that the astrocyte expression of GAD, as the classical enzyme synthesizing GABA, is too low to produce a physiologically relevant amount of GABA. However, in addition to the GAD-dependent pathway, GAD-independent pathways for the synthesis of GABA have been described. Cultured astrocytes are able to synthesize GABA from the polyamine putrescine, thereby utilizing a GAD-independent pathway of GABA synthesis. Recent studies performed both in brain slices and in vivo confirmed the capability of astrocytes to synthesize GABA in both GAD-dependent and GAD-independent ways, and moreover, that the amount of synthesized GABA depends on physiological/pathophysiological conditions. As astrocytes express aquaporins, their membrane is permeable for water. Osmotic gradients between the extracellular and intracellular solutions drive water fluxes across the cellular membrane, influencing the cell volume in this way. An orchestrated functioning of ionic channels and transporters is required to keep the cell volume unchanged or allow controlled cell swelling or shrinkage. Several volume-gated ion channels contribute to the volume control of astrocytes. Volume-regulated anionic channels are outwardly rectifying anion channels activated by cell swelling, encoded by the SWEL1 gene and permeable for larger anionic or uncharged molecules such as glutamate or GABA. Bestrophin are swelling and calcium-activated chloride channels, BEST1-4, with over 200 BEST1 gene mutations linked to retinal diseases. BEST1 is highly expressed in neurons and astrocytes of the hippocampus, thalamus and cerebellum. It is permeable to GABA, lower than for glutamate, 0.47 to 0.67, but this estimate may be low since GABA is mostly uncharged at physiological pH, requiring further study. Blocking or silencing BEST1 reduces tonic GABA currents in cerebellar astrocytes by 75%, showing Bergman glia and lamellar astrocytes release GABA via BEST1 at rest. BEST1 is also abundant in thalamic nuclei. Thus, in regions with high astrocytic GABA, cerebellum, thalamus, tonic BEST1-mediated GABA release occurs under resting conditions, modulated by intracellular Ca2+ and GABA gradients. The exact GABA permeability and selectivity over glutamate need more research. Low intracellular GABA may cause BEST1 to relocate near glutamatergic synapses, switching released glia transmitter to glutamate. In the classical view, GABA transporters JTs remove GABA from the extracellular space. During JT-mediated uptake, one GABA molecule is co-transport with one chloride ion and two sodium ions. Because GABA is mainly a zwitterion at physiological pH, this transport process is electrogenic. Studies have found that the GAT transport process can reverse, known as the reverse mode, releasing GABA instead. This reversal happens when the membrane potential is more positive than the GAT reversal potential, which can occur due to membrane depolarization or an increase in intracellular sodium concentration. Indeed, depolarization-induced GAT-mediated GABA release has been reported in retinal horizontal cells, neuronal growth cones and various neurons in culture or acute brain slices. The reversal potential of GAT in astrocytes can be estimated using a formula. Extracellular GABA concentration varies significantly across different brain regions. Using these data, the intracellular GABA concentration required to set the GAT reversal potential close to the resting potential of astrocytes can be estimated. These estimates suggest that GAT reversal is more likely to occur in the cerebral cortex under resting conditions. Moreover, the relative position of the reversal potential as well as the direction and strength of tonic GABA release from astrocytes can be modulated by transient changes in intracellular sodium concentration, for example, through activation of excitatory amino acid transporters, AATs. In summary, tonic GABA release mediated by GAT3 in astrocytes mainly occurs in the cerebral cortex, likely due to the low extracellular GABA concentration in this region. In areas with higher extracellular GABA, such as the hippocampus, an additional increase in intracellular sodium concentration is needed to reverse GAT3-mediated GABA transport. Additionally, intracellular calcium levels affect the number of GAT3 transporters on the plasma membrane, thereby regulating the strength of GAT3-mediated GABA release or uptake. Alzheimer's disease (AD) is a neurodegenerative disorder with increasing prevalence in the elderly. Evidence shows that aggregation of amyloid beta (A-beta) peptide and tau protein triggers pathological changes, including weakened synaptic transmission and synapse loss. Astrocytes, as part of the tripartite synapse, are involved in AD pathology. Some studies suggest that GAT3-mediated GABA release underlies the enhanced tonic inhibition observed in these animals. In the hippocampus of another AD mouse model, AP/PS1 mice, GABA release from reactive astrocytes has also been reported. Pharmacological studies show that the increased GABA level in astrocytes is due to elevated monoamine oxidase B activity, confirming that GABA is synthesized from putrescine. Additionally, JT1 and JT3 expression in AP/PS1 mice is not significantly changed. It has been demonstrated that reactive astrocytes release GABA via BEST1 channels. Moreover, the membrane distribution of BEST1 in reactive astrocytes of AP/PS1 mice is significantly altered, mainly localized to the soma and proximal dendrites, rather than the distal microdomain-like distribution seen in wild-type animals. These findings contradict reports of GAT3-mediated GABA release by other researchers, and the exact reasons require further investigation. Since the increase of GABA in reactive astrocytes shows a bell-shaped relationship with A-beta plaque accumulation, it is suggested that both astrocytic GABA levels and active release mechanisms may depend on the disease stage, similar to data reported in epilepsy. Huntington's disease (HD) is a neurodegenerative disorder caused by polyglutamine expansion in the huntingtin protein. Previous studies reported decreased levels of excitatory amino acid transporters (ATs) in astrocytes, suggesting that elevated extracellular glutamate concentration and excitotoxic cell death may contribute to HD symptoms. Besides glutamatergic dysfunction, GABAergic signaling, including tonic GABA receptor-mediated inhibition, is also disrupted. JT1 is the main GABA uptake mechanism in the striatum, and pharmacological blockade of JT1 may affect the direction of JT3-mediated GABA transport. Studies suggest that in the healthy striatum, AAT-mediated glutamate uptake raises intracellular sodium, keeping JT3 in the reverse release mode. In HD astrocytes, reduced glutamate uptake and lower intracellular sodium prevent JT3 reversal, keeping it in the uptake mode, leading to hyperexcitability of striatal output neurons (SUNs). Experimental data show that GABA release from striatal astrocytes is reduced in HD. However, the exact cause of this reduction remains unclear. Autism spectrum disorder, ASD, is a neurodevelopmental disorder characterized by repetitive behaviors, communication deficits, and sensory abnormalities. The cause of ASD remains unclear, but the main hypothesis is that an imbalance between excitation and inhibition E/I leads to the symptoms. An increased E/I ratio may result from changes in glutamatergic and/or GABAergic signaling. Besides enhanced glutamatergic synaptic input, elevated extracellular glutamate levels may also cause excitotoxicity in ASD. Similar to the excitatory effect of glutamate, A reduction in extracellular GABA levels may also shift the E/I ratio toward excitation. Epilepsy is a disorder characterized by excessive network activity. It is generally believed that a shift of the excitation/inhibition E/I balance towards excitation underlies the occurrence of spontaneous network discharges, i.e., epileptic seizures. Pharmacological enhancement of GABAergic inhibitory transmission helps suppress neuronal hyperexcitability and prevent epileptic seizures. In this context, GABA release from astrocytes and the resulting tonic GABA receptor-mediated inhibition may be an important means to restore the imbalanced E/I. In a low-magnesium-induced epilepsy model, JT3-mediated GABA release significantly shortens seizure-like events, confirming the inhibitory role of GABA release from astrocytes. The results indicate that epileptic activity stimulates both GABA synthesis in hippocampal astrocytes and GABA release from astrocytes. Whether the GABA release is triggered by EAT-mediated intracellular sodium increase causing JT3 reversal, or mediated through BEST1 channels, requires further experimental verification. We have summarized experimental results on GABA release under physiological and pathophysiological conditions. Increasing evidence shows that astrocytes can synthesize GABA through different metabolic pathways. In the cerebellum and thalamus, GABA is mainly released via the BEST1 channel, while in the cerebral cortex and striatum, JT3-mediated GABA release predominates. Under disease conditions, GABA release from astrocytes is typically enhanced. In some cases, BEST1-mediated release is potentiated, while in others the GAT3-mediated pathway plays the dominant role. Why nature selects one release pathway over the other remains unclear. Elucidating the mechanisms regulating GABA release may help develop effective tools for treating pathophysiological conditions. Thank you for watching. If you want to gain more knowledge, subscribe to the Creative Diagnostics channel. We are dedicated to help you succeed and advance your research progress. Visit our website for more information.